Welcome back, beta. Today we will discuss about the Hybridoma technology. This is the new headings or new contents that we have to learn. So in these contents, some of the basic things are required. For example, what is hybridomas, what is the application of hybridoma technology, and what are the meaning of epitopes, paratomes. So let us discuss about the Hybridoma's technology. Hybridoma technology is the technology that was developed by two great scientists, that is Kohler and Milstein. So Kohler and Milstein is the two scientists who got the Nobel Prize for the discovery of this hybridoma technology, and the product of hybridoma technology is hybridomas. Hybridomas is the product of hybridoma technology. Now let us see what is this product and how it is very important. Actually, when this Kohler and Milstein was working on this technology, on this project, so it brought the revolution in science and technology to find out the diagnosis of the various kind of those diseases which are becoming fatal for the human society. So before coming to this hybridoma technology, let us see what is the meaning of epitopes. What is the word epitopes? Epitopes determine the particular specific part of antigen. You can see that this is the structure of the antibodies, and you must know that antibodies have the general formula H2L2. H stands for heavy lines or heavy chains, and L stands for the light chain. So each antibodies is made up of two light chains and the two heavy chains. So you can see that this is a particular part. Here what happened, any antibodies are attached. So this is the place where antigens or specifically bind here. So what is the meaning of epitopes? It is the antigen specific part which binds on an antibodies. Now the second word is that epitomes or epitomes. The meaning of epitomes are, this is the place or it is the antigen antibodies complex structure. Suppose here I just settle down here, this red dot is the antigen. Okay, and this total structure is the antigen antibody complex structure. So antigen antibody complex structure is known as epitomes. Now the word is what? Another word is paratopes or paratops. These paratops is likely the specific part of antibody. Suppose again I am drawing this. This is again I draw the antibodies structure. Suppose these are the part of antibodies where any antigens can bind or specific antigens are bind to this location. So this location is known as paratopes. So paratopes are the antibodies specific part or domain where any antigens bind. So as we know that these blood cells, particularly the serum, you know the RBC, WBC, these are the fluid connected tissues, and here the more important thing is that the serum. What is that? Serum. And out of the serum we are taking the white blood cells, and these white blood cells are polyclonal antibodies. Again here we will discuss about the differentiation between the monoclonal antibodies and polyclonal antibodies. So as we know that this serum is having the B cells, that is B lymphocyte cells, and these B lymphocyte cells are the mixture of various B cells that producing the different kind of antibodies or different forms of antibodies. Several antibodies are produced from the B cells of the serum. So it is known as polyclonal antibodies. Poly means many, clonal means various forms. So various forms of the antibodies which are taken out from the B cells, that is polyclonal antibodies. Now the monoclonal antibodies means here the production of homogeneous type of population of antibodies that is taken from the B cells. It means a B cells is taken out from the serum, specific B cells are taken out from the serum, and from which we are making one kind of the identical kind of the antibodies. So it is known as monoclonal antibodies. Polyclonal antibodies and monoclonal antibodies have again some variation. These polyclonal antibodies are the heterogeneous population of the antibodies, whereas monoclonal antibodies are the homogeneous population of the antibodies. It requires hybridomas. Actually, monoclonal antibodies can only be produced by the hybridomas technology. So let us see what is the meaning of this hybridoma technology and how it is produced. Here we are taking the mouse and we are having the myeloma cells. Myeloma cells are the cancerous cells. These are the cancerous cells. And here we are taking the lymphocyte cells. And you know the lymphocyte cells are the production of the B cells. They are the immunity booster, immunity providing substance that we call the antibodies. So we are taking the mouse and spleen is taken out. Why we are opting the spleen only? As we know the spleen is the graveyard of RBC, and here we know the RBC are down. So we are taking out the spleen cells. So children, we are taking these are the spleen cells. Okay. After taking the spleen cells, it is now fused with myeloma cells, that is the cancerous producing cells. What happened when these spleen cells and myeloma cells both are fused under PEG, that is called polyethylene glycol. It is required for the fusion of the cells. So this mixture is known as hybridomas. So hybridomas is the mixture or combination of spleen cells that containing the B cells and the myeloma cells, that is the cancerous cells, and it is worked out or it is done in the medium that is called polyethylene glycol. So this is the hybridomas. Now these hybridomas are further processed or cultured in a medium that is called hat medium. Hat medium has very good importance, or they have some superficial importance in the culturing of the monoclonal antibodies or the hybridomas technology, because hat medium is responsible to promoting the continuous cell division, uncontrolled cell division, and it is equally responsible for stopping or it sees the de novo synthesis of the DNA. It favor or it promote the continuous or diffuse cell division, so that the number of myeloma cells and the spleen cells that contain the B cells profusely divide and their population increases. So we prefer to take the hat medium. What is the hat medium? It stands for hypoxanthine aminopterin thymidine. Again I am repeating, hat medium is a medium which is especially taken for the culturing of mammalian cells and production of infinite number of cells, and it stands for hypoxanthine aminopterin thymidine. Overall, that from the hat medium we have to segregate and harvest the monoclonal antibodies, that is called the MAPs. Now these MAPs which is taken out from the hat medium are used for the detection of those fatal diseases like cancer and the AIDS in the early stages of the disease spread. So this is about the hybridomas technology. Once again, I am repeating the nutshell. Hybridoma technology was developed by the two biologists, two scientists who got the Nobel Prize, and they were Kohler and Christine, Nobel Prize winner, and they have produced the hybridomas. What is the hybridomas? Hybridomas are the fusion or the combination of the spleen cells that containing B cells, and another one is the myeloma cells that is responsible for the cancerous cells or cancer producing cells. Both are fused in a medium that is called PEG, polyethylene glycol. Again from there, this mixture hybridomas are further processed or cultured in the head medium. We are opting the head medium because it promotes the continuous cell division and stop the de novo synthesis of the DNA. And from there, we are taking out the harvesting the maps, and that maps are used to earlier detection of the fatal diseases like cancer and AIDS. Again, that I just told about the epitopes and paratopes. This is the structure of antibodies, and these antibodies is having the H2 L2 chain, that is 2 heavy chain and 2 light chain. So the specific part of antibodies where antigens bind is known as paratopes. Antigen and antibodies complex structure is known as epitopes. Epitomes. And this last word, epitope, is the specific part of antigens which take part in the antigen antibody interaction. Thanks for watching.